Hi everyone, welcome to my A-level biology lecture series. In this lesson, we will explore oxidative phosphorylation. At the end of this lesson, you will be able to explain that during oxidative phosphorylation, hydrogen atom splits into proton and energetic electron. Energetic electron release energy as they pass through the electron transport chain. The released energy is used to transfer proton across the inner mitochondrial membrane. Proton return to the mitochondrial matrix by facilitated diffusion through ADP synthase, providing energy for ADP synthesis. Let's get started. Oxidative phosphorylation. Oxidative phosphorylation is the final stage of aerobic respiration where energy released from the electron transport chain is used for the phosphorylation of ADP to form ATP. Oxidative phosphorylation occurs at the inner mitochondrial membrane. So the key process in oxidative phosphorylation involve electron transfer in the electron transport chain, as well as chemiosmosis. So this diagram shows the overall process of oxidative phosphorylation. So the entire process of oxidative phosphorylation involve two main stages. The first stage is known as electron transport chain, and the second stage is known as chemiosmosis. So you can see that we have electron carrier here to form electron transport chain. So a complete electron transport chain consists of a series of electron carrier. So chemiosmosis involves an enzyme known as ATP synthase. So how oxidative phosphorylations occur? So first of all, we refer or we take note on this reduced NAD. So reduced NAD release hydrogen atom, and this hydrogen atom split into protons and electrons, and this electron has high energy level. So these electrons flow through a series of electron carrier which form electron transport chain. So when this electron passes through the electron transport chain, energy is released. So this energy is used by this electron carrier to transport proton from the mitochondrial matrix into mitochondrial intermembrane space. So you can see that from this diagram, when electron pass through an electron's carrier, proton is pumped into mitochondrial intermembrane space. So overall reactions, what will happen here is because the transportations of proton from the mitochondrial matrix into the mitochondrial intermembrane space against the concentration gradient. So there is an accumulation of protons at intermembrane space, and there is a lack of protons at mitochondrial matrix. So a steep proton concentrations gradient is created. So, however, you can see that in these parts, because due to the accumulation of the positively charged proton and lack of the positively charged protons at the mitochondrial matrix, so we can actually describe this gradient as electrochemical gradient due to the uneven distributions of charge. So for protons to come back or to diffuse back into the mitochondrial matrix, it's impossible for proton to pass through directly or diffuse across the mitochondrial inner membrane because it is a barrier, because of this phospholipid bilayer. The only way for protons to move back from the mitochondrial intermembrane space back to the mitochondrial matrix, they have to pass through this proton channel. So when they pass through this proton channel, energy is released, and this energy will be used by this ATP synthase to phosphorylate ADP to form ATP. So you can see that the entire process of oxidative phosphorylation, okay, require reduce NAD and reduce FAD to release hydrogen atom. So basically, they carry hydrogen atom from link reactions, from Krebs cycle, and then pass these hydrogen atom to electron transport chain. So when they donate this hydrogen atom, so hydrogen atom splits into protons and electron. So the electron flow through this electron transport chain and finally accepted by oxygen. So now, what is the role of oxygen in oxidative phosphorylation? Oxygen actually acts as final electron acceptor. So when this oxygen accept electron together with the proton, so oxygen now is reduced to form water molecule. So again, this is a redox reaction, oxidative and reduction. So oxygen is reduced to form water. At the same time, reduced NAD is oxidized back to form NAD. So oxidative phosphorylation actually has two roles. First, to synthesize ATP. The second role is to regenerate back NAD as well as FAD. So as we know that the entire process of oxidative phosphorylation consists of two main stages. So the first stage is transfer of protons and electron. So first of all, reduce NAD and reduce FAD donates hydrogen atom. Be careful, hydrogen atom, not proton, okay, to this electron transport chain. And this hydrogen atom splits into protons and electrons, and this electron has high energy level. So these electrons are transferred through a series of electron carrier that form electron transport chain. So at each level, okay, at each electron carrier, energy is released. And this energy is used to pump protons from the mitochondrial matrix into the intermembrane space, creating a steep proton gradient or a steep electrochemical gradient. So these four processes belongs to the electron transportation, okay, along the electron transport chain. So following this process is chemiosmosis as well as ATP synthesis. So proton now move back into the mitochondrial matrix by facilitated diffusion through a proton channel, which associated with ATP synthase down the proton's concentration gradient or electrochemical gradient. So as protons flow down their concentration gradient from high potential energy to low potential energy, definitely energy is released. So this energy is released, the energy released here is used to phosphorylate ADP to form ATP in a process known as chemiosmosis. So at the end of this electron transport chain, oxygen acts as the final electron acceptor by accepting accepting proton and also electrons. So this oxygen is reduced to form water. So what is the role of oxygen in aerobic respiration? We always say that oxygen acts as the final electron acceptor during oxidative phosphorylation. The oxygen combined with proton and electron to form water as the byproduct. So this process is an example of oxidation reduction reactions, or we call it as a redox reaction. So oxygen is reduced to form water molecule by gaining the electrons combining with the proton to produce water molecule. On the other hand, reduced NAD and reduced FAD are oxidized as they donate hydrogen atom. And this is very, very important because this oxidation allows the regenerations of NAD and FAD. So with sufficient NAD and FAD, Krebs cycle and link reactions can continue. So what will happen if oxygen is absent or without oxygen? So first of all, without oxygen, the electron transport chain stop functioning. This is due to without oxygen act as a final electron acceptor, reduce NAD and reduce FAD, will not release hydrogen atom. Okay, because we know that redox reaction always happen together. Because oxygen is absent, so without oxygen to act as a final electron acceptor, reduce NAD, reduce FAD are not going to release hydrogen atoms. So without reduce NAD and reduce FAD to release hydrogen atom, there's no electron to pass through electron transport chain. So therefore, electron transport chain stops functioning. So when electron transport chain stop functioning, so proton gradients cannot be generated. So without a steep proton concentration gradient, definitely ATP synthesis by chemiosmosis stops. Next, without oxygen, link reaction and Krebs cycle stop as well. Why? Because without oxidations of reduced NAD and reduced FAD, so there is no or insufficient NAD and FAD to continue the link reaction and Krebs cycle. So what is the next consequences? Next consequence, you can see that pyruvate cannot enter into the mitochondria because there's no point for pyruvate to enter into the mitochondria because link reaction can't take place, Krebs cycle can't take place due to the low supply or insufficient NAD and FAD. So as a result, the cells now have to switch from aerobic respiration to anaerobic respiration, where pyruvate will be converted into lactate in animal or mammalian muscle cells, or ethanol and CO2 in the yeast and plant. So due to this anaerobic respiration that take place in the cytoplasm, so NAD can be regenerated for glycolysis to continue so that a small yield of ATP can be produced. We are going to look at this anaerobic respiration in other lesson. So now let us compare the ATP yield for the complete oxidation of glucose. Let us look at the ATP produced by substrate-linked phosphorylation during glycolysis. There is a net gain of 2 molecules of ATP. Link reaction, unfortunately, no ATP is produced through substrate-linked phosphorylation. Krebs cycle, yes, we have 2 molecules of ATP produced through substrate-linked phosphorylation. So there is a total of 4 ATP produced by substrate-linked phosphorylation. By looking into this figure, we will know that 4 ATP molecules is insufficient for the cells to sustain. So therefore, we need more ATP to be produced through oxidative phosphorylation. And oxidative phosphorylation actually depends on the number of reduced NAD and the number of reduced FAD. Let us look at how many reduced NAD and reduced FAD can be produced through aerobic respiration. During glycolysis, there are 2 reduced NAD produced through glycolysis, another 2 molecules through link reaction, and 6 molecules through Krebs cycle, making it a total of 10 molecules reduced NAD produced through the aerobic respiration. How about reduced FAD? Reduced FAD can only be produced in Krebs cycle. So there is a total of 2 reduced FAD per complete oxidations of glucose. So if you look at this figure, 10 plus 2, there is a total of 12 molecules of reduced NAD and reduced FAD. And this corresponds to the 12 hydrogen atom available in a glucose molecule. So now we want to know how much or how many ATP can be produced through oxidative phosphorylation. So on average, each reduced NAD contributes to a synthesis of 2.5 ATP molecules, and reduced FAD contributes to the synthesis of 1.5 ATP molecules. So by a simple calculation, we know that there is a total of 10 reduced NAD produced through the oxidations of glucose. So each reduced NAD contribute to 2.5 ATP yield per molecule. So therefore, 25 ATP molecule can be produced or can be synthesized through this oxidative phosphorylation through the contributions from reduced NAD. For reduced FAD, there's a total of 2 molecules per glucose molecule. So each reduced FAD contributes 1.5 ATP molecules. So therefore, a total of 3 ATP can be synthesized. So therefore, in total, there are 28 ATP molecule averagely can be synthesized through oxidative phosphorylation when we completely oxidize a molecule of glucose. So let us total up the entire ATP yield. First, we know that substrate-linked phosphorylation produces 4 ATP molecule. Oxidative phosphorylations on average 28. Therefore, there is a total of ATP. So there's a total of 32 ATP molecule per glucose molecule. Can be synthesized. However, some textbooks suggested that a maximum ATP yield of 38 ATP per glucose molecule, with the assumptions that each reduced NAD generate 3 ATP and each reduced FAD generate 2 ATP. However, a more widely accepted value in the modern biology is 32 ATP because there is energy loss. Okay, so with the idea of 38, that is a maximum number. Consider that or assume that there's no energy loss, but it is impossible because energy loss always takes place due to not all energy from electron transfer in the electron transport chain is used to pump protons. As we know that when electron passes through this electron carrier, energy is released, but not 100% of energy is used to pump proton. And some proton actually leak back into the mitochondrial matrix without passing through ATP synthase. We do know that phospholipid bilayer act as a barrier for the proton, but somehow some proton can actually leak back into the mitochondrial matrix without passing through ATP synthase. So therefore, this energy loss take place results in a lower number of ATP yield, okay, compared to the theoretical value, which is 38 ATP. And some ATP must be used, okay, to transport the pyruvate from the cytoplasm into mitochondrial matrix. With that, we have come to the end of this lesson. I hope you have found this lesson helpful. If you have any question, again, feel free to leave them in the comment sections. See you in the next lesson. Have a great day.